So we're going to start with COVID-19, and it really has been a new type of pandemic, especially in Africa. Our unique genetic diversity, the size of the continent, the disparities in our climate have meant that the pandemic has taken a very different form in Africa, so much so that there are a number of studies trying to understand why Africa, which was always predicted to be so much more at risk than anywhere else in the world, seems to have been able to come through the majority of the pandemic much better than some of their much more technologically advanced counterparts. But the cost of Africa remains incalculable if we cannot find our own solutions. And that's really what this pandemic has shown us. The global community is not equipped to help Africa solve her problems. And we have to start building capacity locally that we can solve our own problems. And one of the key things to making sure that Africa is able to solve her own problems as a continent is to prioritize the health system capacitation, especially in the face of a global pandemic, recession, and humanitarian crisis. We have our own realities here, and those realities are not necessarily taken into account by our global counterparts. So it's really important for us to start thinking about ways that we can work to get our own solutions to force the healthcare of Africa to improve and to just capacitate our ability to service our communities better. So if we look at the COVID-19 map, we can see, of course, the United States famously have failed to control the coronavirus, and most of Latin America is also experiencing extremely high numbers of infections. Africa, despite the pundits saying that we were going to be in so much trouble with this virus, we've actually been able to hold on to our public health pretty well. And I think one of the reasons is that we have a lot of experience in infectious diseases. So although coronavirus itself is completely unique to us, we've had outbreaks of diseases like Ebola, we have influenza, diphtheria, tuberculosis, and those have all taught us truly how important isolation, tracking and tracing events are, and we've been able to use that knowledge to sort of minimize the effect of the virus. We are underreporting in Africa, and that's because of constraints around technology and also in terms of availability of reagents. And one of the things that this pandemic has very much shown us is that Africa has to be able to develop their own diagnostics, develop their own therapeutics, develop their own vaccines, because we cannot rely on Western Europe and North America for their support. So when the African study, the Center for Strategic Studies looked at Africa as a whole in terms of the pandemic, they sort of rated on how well our healthcare systems would be able to cope with this. And you can see that for the most part, Africa has relatively under-resourced healthcare systems, especially in our larger population areas. South Africa has a famously weak public healthcare system just because it's deeply under-resourced in terms of the size of our population. And that led to the sort of worst case predicted deaths for coronavirus. And you can see that Nigeria, Ethiopia, and South Africa all have 200,000 or more predicted deaths if there were no interventions managed in those regions. And that's really still a worst case scenario for us. What we've seen in Africa is not the steep peaks and valleys that we're seeing in sort of more developed regions. What we're seeing in Africa is a really prolonged curve. And that's because we've been able to implement these sort of localized knockdowns, and our people have been very compliant. But what we're seeing is this sort of extended public health crisis. And what that means is that although we haven't had sort of an overcapacitation of our hospitals and so on, what we are seeing is this sort of really extended viral lifespan in our population. And part of that is associated with this figure, and that is that our healthcare systems are just not as well equipped. So if you look at just hospital beds per 10,000 inhabitants, South Africa is probably the highest number, and we're close to the UK, and we have a population of only 54 million. As we move into the rest of Africa, you can see that number of beds per 10,000 inhabitants really drop. And what that means is that our critical healthcare systems are not built to withstand really large numbers of infections. And that means that it becomes really important that we find other solutions. And one of those is to introduce vaccines into our populations that prevent the need to go to hospital. And we also have to take into account that the pictures on the right-hand side are a reality for large portions of the continent. We tend to be quite rural. We have areas of very highly densely populated cities, and then often our medical professionals are working in conditions that are less than ideal. And all of that sort of combines to mean that Africa's healthcare system is very, very different. And what we need to do is make sure that our healthcare system is improved, obviously, but more importantly than that, we need to start looking at preventative measures so that we're ready for the next pandemic, because the science says we will have another one. And we've still got probably another 2 years of coronavirus ahead of us. So we have to start thinking about how do we build our systems to be more robust to withstand these sort of impacts. So what is coronavirus? Well, we know coronavirus, or COVID-19 as people refer to it. So COVID-19 is the disease and coronavirus is the infectious agent. Coronavirus is closely related to severe acute respiratory syndrome, or SARS, and we talk about SARS-CoV-2. It causes adult pneumonia, similar to diseases caused by influenza virus, adenovirus, parainfluenza virus, and probably the most similar in terms of pathology is the respiratory syncytial virus. We know that there's this increased susceptibility of immune suppressed hosts, which I think is one of the reasons that we were so critically concerned around Africa with our high degree of HIV infection. We were worried about comorbidities and what that impact would be on coronavirus. Coronavirus preferentially infects the alveolar epithelial cells. And that's because these cells express the most highly express the ACE2 receptor, which is what coronavirus uses to enter the cells. And what we know additionally is that there's a subgroup of patients who develop severe COVID-19 who experience a response called cytokine storm. And cytokine storm syndrome is just simply an overactivation of the immune system that causes sort of systemic damage and shutdown. So that's coronavirus, and that's what we know about it. At a molecular level, we actually know quite a lot about COVID-19 already, and that's probably because we've been able to use things like next generation sequencing technology really effectively in this outbreak. And what we know about it is, of course, it's a coronavirus. It is a positive single-stranded RNA virus. It has a genome of around 30 kilobases, and its variant is between 50 and 200 nanometers in diameter. Probably most importantly, from a vaccinology point of view, we know it has 4 structural proteins, and we know that the spike protein in particular is antigenic, which means it develops antibodies against it, and that the spike protein is reasonably accessible. Okay, it's a pretty simple protein, and what that means is that if we can target that spike protein, we may be able to inhibit, coronavirus. We can really create a robust immune response, which is what you need for a vaccine. So if we look at the SARS-CoV-2 life cycle, we know that it interacts with ACE2 and TMPRSS2 to enter into cells, where it's obviously unpackaged, and our RNA genome undergoes replication, exported to the Golgi where it's repackaged, and then excreted to the other cells. And this is a really, really efficient system. And there are quite a lot of studies for therapeutics that focus on inhibiting the Golgi expression, inhibiting ACE2 interactions, inhibiting TMPRSS2. But from a vaccinology point of view, our interest is primarily in the proteins on the surface, or the cellular epitopes that we can identify for SARS-CoV-2 and then emulate in a vaccinology program. So are vaccines the answer? I mean, we keep talking about vaccines and therapeutics hand in hand, and there's always this question, are vaccines really, really what we should be talking about? And I'm going to use a little bit of data, but I'm going to say yes, vaccines very definitely are the correct response to pandemic. And critical to future pandemic preparedness is our ability to create and rapidly produce vaccines on the continent. And that's just because if you think about the manufacturing scale, you're talking about billions of doses of a vaccine at any given point in time. And there is a global constraint on manufacturing for vaccines, and we'll talk about that in a minute. So a vaccine, I'm sure you all know, but just to make sure that we're all on the same page, is a biological preparation that provides active acquired immunity to a particular infectious disease. Okay, so vaccines are disease specific, and they're designed to prime the immune system. So a vaccine typically contains an agent that's similar to the disease-causing microorganism that stimulates the body's immune system, and particularly the memory response. So what we want from a vaccine is to teach our body to recognize the invader. So we want our vaccine not just to have a localized inflammatory response, but it's really, really important that our vaccine induces the production of our memory B and T cells. It's really, really important for proper immunization that occurs. And it also, one of the reasons why some vaccines require multiple doses is because that memory is not long-term. So your influenza vaccine is a really good example. Not only do we get a lot of mutational changes one season to another for influenza, but over time, our body purges those memory cells to make space for new ones. And so it's really important to keep vaccinating yourself. So it's the same, probably for coronavirus, we're going to have this novel iteration after iteration of vaccine. And that's another reason why it's really important that we start developing our own vaccines locally. Vaccines can be used prophylactically, so that's prevents future disease, which is the context that we're discussing today. But vaccines can also be therapeutic. So there are lots of firms and research groups that focus on the development of therapeutic vaccines, especially oncolytic vaccines that target cancer cells. And it is something that Merck also has a lot of experience in. So if you're interested in that kind of vaccine, please reach out to us, but we're not going to touch on that today. Today, we're talking specifically about prophylactic vaccines. So are vaccines useful? I think we all know that they are, but just so that we can put it into context. Smallpox was the first vaccine, and it basically changed the whole shape of how we deal with pandemic and endemic antigens. So vaccines have greatly reduced the prevalence of disease everywhere in the world. And we've seen an improvement in vaccine coverage over the past several decades, but there's still 13.5 million children who were not vaccinated in 2018. And that's really a tragedy, because vaccines are the most successful in a cost-effective way to prevent disease. So increasing vaccine coverage leads to the decline of diseases. It's indisputable. And that means that capacitating and producing more vaccines is absolutely critical to long-term health and prosperity. So 9 out of 10 people in the world think that vaccinating children is important. And we really are, our coverage of vaccines remains way, way too low. So we're really missing out on an opportunity to save so many more lives. Vaccination relies on this idea of herd immunity. And vaccines really accelerate herd immunity. You can get to natural herd immunity, and we'll talk about that in a minute. But what vaccines do is they really accelerate herd immunity without getting a lot of sick people. So natural herd immunity relies on a large proportion of your population becoming ill and then becoming immune, so developing antibodies. Takes a long time, and you get a lot of infected people. What immunization allows us to do is to create an artificial exposure program. So instead of becoming sick, these people simply produce antibodies to the antigen. And that means that they're not going to get infected. And then they provide coverage and protection for people who can't receive the vaccine for some reason, or who are too young for it, etc. And what that does is it really protects our communities as a whole. So immunization via vaccines accelerates herd immunity and prevents communicable diseases with significantly reduced risks to human health. And that's really where the importance of vaccines becomes most obvious. So if we talk about herd immunity, what kind of percentages are we talking about in terms of population? When is our population really going to have the full effect of a vaccine? So if we look at a disease like measles, which is airborne and has a really, really high reproduction rate, it's airborne. Measles is probably one of the most infectious diseases in the world. We're talking about herd immunity threshold of around 92 to 95%. And that's really why we've seen this sort of resurgence in measles in response to anti-vaxxers. We've gone from being primarily immunized against measles to having people who are selectively not immunizing, and we're dropping that herd immunity threshold every time, and that's allowed this resurgence of this disease. So measles is like a really, really extreme example of what can happen if you don't vaccinate. But if we move down through some of the less potent viruses and we come closer to what we look at for SARS-CoV-2, if we look at SARS, for example, it's an airborne droplet disease. Its basic reproduction number is somewhere between 2 and 5, which means our herd immunity threshold is 50 to 80% of the population. Okay, so it's a huge number. And if we look at the antibody estimates from some of the most infected countries in the world, in early May, we were looking only at best 20% of the population had exposure and had developed antibodies. We need 3 times as many, and it's an exponential number in terms of covering our population. So you can see that natural herd immunity to this virus will take several years, if not decades. And that's obviously inhibited also by all of the interventions that we've taken to sort of stop the spread of COVID-19. And if we look at what happened in Sweden, where they didn't go into lockdown, they haven't reached herd immunity either. So it's really important to recognize that herd immunity, natural herd immunity, is very, very hard to reach. It requires really a very, very high number of infected people. And it puts a huge burden on healthcare systems. So it's really important to develop ways to rapidly develop our immunization programs and really put them into place as fast as possible. One of the major things that we have to consider coming into the COVID-19 vaccines is that vaccine requirements worldwide were already increasing before we encountered this pandemic. So if we look at global vaccination coverage, we were talking about 89% for tuberculosis, 86% for measles, 86% for DTP. And you can see that if you think about the world's population, that's really a lot of vaccines to be manufacturing. So there's a complex set of factors, including bioproduction and cost limitations, that have limited our ability to provide critical vaccines to many economically challenged countries. And that predominantly affects our region of the world, right? Africa and the Middle East. And what that means is that those economically challenged countries, or those countries, have got to start developing their own manufacturing potential, because as the Western world, as North America and Western Europe emerge from COVID and they get ready for their vaccines, they're going to be taking a lot of these first line vaccines offline because they have to make the manufacturing space for the new ones. And then countries in Africa where we have this high density of tuberculosis and hepatitis, we're suddenly going to have so many fewer doses. And that's really where the critical crunch is going to come from. So not only do we need to look at building our capacity for making COVID-19 vaccines and pandemic vaccines, we also need to invest in our ability to produce and manufacture vaccines that we need locally, and technology transfers for those vaccines. So it's really, really important that we think not just about COVID-19 in terms of vaccines, but vaccines in general and the applications that we need to start looking at in terms of growing our capacity for them. So the problem with Africa and vaccines is that vaccine manufacture is a complex multi-step process. You have to first manufacture the antigen, you have to grow the organism, you have to inactivate it, and you have to purify it. Then you have to formulate and fill. So you have to blend the vaccine, fill the vaccine, and freeze dry it. Then you have to distribute and package. Okay, and all of those things are complicated by the number of countries, the size of our continent, the climate. Cold storage in Africa is a real concern. So you have to think about all of those things when talking about vaccines in an African context. And each step has specific equipment, facilities, skills related to each vaccine. And it's really, really important to remember that we don't, as a rule, have a lot of that experience in Africa. So if we look at the world vaccine producers, you can see most vaccines are produced in China, the United States, and Western Europe. And Africa, with the exception of South Africa, there's not a lot of vaccine manufacture for human consumption. Animals are slightly different. But for human consumption, African vaccine production is still quite low. And we're one of the largest users of vaccines in the world. And we add into the fact that there's a prediction that Africa will be home to 25% of the world's population by 2025. So we have a growing population. We have this need to make sure that our healthcare system is more robust and that we're sort of investing in technologies that allow us to build these robust infrastructures. So in 2015, 23 African countries spoke out about the increased financial burden of new vaccines at the World Assembly. And they at the time tried to put pressure on pharma companies to lower vaccine prices. And sure, I mean, there is an argument to be made for that. But one of the most critical ways to do that is to start producing vaccines ourselves. And so it's really important that we start developing the skills to make our own vaccines, invest in the infrastructure. And unfortunately, it's expensive infrastructure. If you can create vaccines, it's an entire market on its own. There's an economic boost to it because vaccines are export product, not only for local consumption. And investing in this kind of technology is investing not only in the health of our countries, but more significantly in the prosperity. So vaccines are not just a vehicle for health prosperity, but economic growth as well, and should be considered in that light when we're talking about making these large investments. So if we talk about Merck and vaccines, Merck is synonymous with vaccines, more or less. There are currently 10 trials for COVID-19 vaccines, and Merck is actively involved in all of them. And the reason for that is we have a long-term commitment to advancing vaccine research and manufacture. And this is really interesting because our life sciences division, our research part of our business, we really strongly believe in vaccines. And for that reason, Merck has pumped time and money into vaccine development sort of programs. So we've done transfer technologies for vaccine manufacturing to a site in Ghana. We're supporting the Jenner Institute, the Baylor partner. We're a partner with Baylor University for neglected diseases. We partner with Janssen and GE. We also are part of the Divine Consortium. So that's to address the low yields and high cost of vaccine purification processes. So very much a focus on improving the technology, which is our part of the business. And we really have, we invested 110 million in a new gene therapy center in California. So Merck is very dedicated to vaccines. And that's really because one of the largest challenges to vaccine manufacture is in of the vaccine. So to be of maximum benefit, vaccines have to be available to the widest population. We know that. Virus vaccines are cheaper to produce, but have very high risks. And attenuated vaccines are more expensive, but safer. Making sort of subunit or attenuated vaccines more attractive is to improve the yield of the bioproduction processes. So Merck in our vaccine workflows focus on manufacturing technologies. So we produce optimized cell culture bacteria, cell culture medias, bioreactors. All of these things are designed to improve cell densities and therefore improve yield. And we have a lot of new purification resins and membranes that really give us higher recoveries and shorter process times. And that's really to help with this economic development behind vaccines. So if we talk about vaccines, let's get down to the nitty-gritty. What is in a vaccine and how do pros and cons of the different technologies? So vaccines really are made up of two really critical components. The antigen, so that's the active component of the vaccine. That's what causes an immune response. It's usually made up of weakened or killed germs, fragments, or substances made by our targets. And then those are what gives us that B cell response, right? That's what the memory is for. But those are often not that immunogenic on their own, right? So you need to boost your immune system and say to, hey, this is what you should be paying attention to. And so we add adjuvants to our vaccines. And adjuvants are basically salts that force the inflammatory response at the site of vaccination. they recruit the immune system to come there, and that's really what facilitates the B cell recognition. And that's really a very simplistic breakdown, but the two primary immuno vaccine and adjuvants. And vaccines, of course, have additional components, so preservatives, that's to protect from contamination, stabilizers, that's to prevent it from sticking to the flask or going off and having strange reactions. Residues, of course, those will be from the manufacturing process. Most commonly in inactivated viral vaccines, we're talking about formaldehyde. And then diluents, saline, water, sodium chloride. Those are just to keep the vaccine isotonic, which is obviously very important, and maintain pH. And then we have surfactants and emulsifiers, that's around cell uptake, and antibiotics in some cases, especially for long-term storage. So these are all other components that go into the vaccine. What I will say is that Merck has technologies that are approved for all of these additional steps, including preservatives and stabilizers. And with our technologies, you're really reducing the amount of residues that are left in your vaccine. So we've discussed how vaccines work. We inject our vaccine into the body. The body creates antibodies to fight the germs we've introduced. These antibodies become memory antibodies. And what that happens is if you ever encounter the actual virus or threat, your body will start producing those memory antibodies and attack that incoming threat. So vaccines can be broken up into several different types of modalities. We have whole inactivated vaccines, probably the most common vaccines in the world, the oldest technology. Take your virus and you heat and activate it, and then you inject it into people. Live attenuated, those vaccines are able to replicate, but they're not as virulent. And then we can enter the subunit vaccines. And subunit vaccines have really come a long way. And they're really the new horizon in vaccine technology, because subunit vaccines are simpler to produce. And also because you can use the same manufacturing pathway for multiple vaccines. And we'll talk about that in a little more detail in a minute. So we've seen a lot of adoption of subunit vaccines in the new rabies vaccine and also in the new influenza vaccine. And of course, the coronavirus vaccines, more than half of them are what are called subunit vaccines. So if you look at live attenuated viruses, these ones should be really, really familiar to all of us. These are vaccines we would all have received at some point in our life. So tuberculosis, the oral polio vaccine, rotavirus, and yellow fever, just to name a few. Inactivated vaccines, also very familiar to us, poliovirus and pertussis. And then subunit vaccines, depending on your age, these are ones that you may or may not have had. So that's your pneumococcal and hepatitis B vaccines. And just to give you an idea that vaccines are not limited in terms of their application based on their technology. It's just really important to understand that each technology has unique pros and cons, and they require unique sort of manufacturing processes. So vaccines may be monovalent or polyvalent, okay? And that just means that a vaccine can target a single strain or a single antigen, for example, the measles vaccine, or it can be polyvalent, which means that it targets more than one strain or serotype, and that's similar to our oral polio vaccines. Okay, so I'm going to talk about some of the major technologies now and the pathways that go into them, some of the considerations, pros and cons for each. This is not exhaustive. For time constraints, I haven't gone into every type of vaccine, just the ones that are currently under sort of evaluation for COVID-19. So live attenuated vaccines have been available since the 1950s. And these vaccines are primarily derived from disease-causing pathogens that have been weakened under laboratory conditions. So LAVs stimulate an excellent immune response because they're very similar to the pathogen, because they have an infection rate that's nearly as good as the wild-type pathogen. And that means that the functional immune system eliminates the attenuated pathogens during their immune response. And sustained infection can result in local lymphangitis or disseminated infection. So that's the risk with live attenuated viruses. You can have them come back. They can mutate and then become virulent again. If the vaccine is sure, you do sometimes get contamination with other viruses. So it was reported about 15 years ago, there was a retrovirus contamination in the measles vaccines. So a lot of people who received the measles vaccines became very, very ill, and it wasn't measles, it was actually retrovirus. And there are potential immunization errors, and that's because these vaccines predominantly have to be kept under cold storage. And if they become inactivated by heat, for example, you can get changes, and then you're going to get a mispriming of your immune system. So they're quite sensitive vaccines. And it's really important that we keep them under cold storage. So that's probably their biggest disadvantage. So if we want to create a recombinant attenuated vaccine, we isolate the virus, we clone its genome, we identify and then eliminate its virulence genes, and we have a viable virus, so it replicates, so you get a strong immunogenic effect, but it's not virulent, can't pass to another person or cause disease, and these can be used as a vaccine. Okay. The advantage, of course, live attenuated viruses are strongly immunogenic, they're reasonably easy to produce, and you get functional clearance. But the disadvantages for live attenuated viruses are quite significant. You have to have cold stage storage, you have a high risk for potential contamination, they are potentially pathogenic, and they can overstimulate the immune response. So you can get really, really sick from a live attenuated viral vaccine. And that's really why we've seen this move away from live attenuated viruses. But this is a simplified workflow, and just so that you can get a sense of the steps, we prepare the medium and the inoculum. We create an environment for viral infection. We then clarify, so that is we remove the dead cells. We inactivate the virus. We filter, concentrate, centrifuge, remove nucleic acid, fragment the viruses, clarify again, inactivate again, send through TFF or tangential flow filtration, then it goes through sterile filtration, and then it goes to fill. So you can see there are a lot of steps, it's quite complicated, and you can see that the more steps you have in a process, the more points of failure you're going to have in a process. And you can see every single step also introduces a potential for contamination. So you can see that live attenuated viruses, although they're very immunogenic, are not necessarily the number one choice for modern vaccines. And that's just because there's so many disadvantages. And you can see this is a very long process, and each of these processes is entirely individual for each virus. So they're not steps you can share between different vaccines. These are all steps that have to be done individually every time for different viruses. You actually have to have completely different lines for this kind of vaccine production. So it's a really important consideration if what you're looking for is an investment in a technology that allows you to do more than one type of vaccine, right? Then we come to inactivated viral vaccines. So these are whole cell vaccines that have been killed through physical or chemical processes, predominantly used for bacteria. They can't cause disease. They don't always produce a very strong immunogenic effect. And that's what we've seen with some of the first round COVID-19 vaccines. We saw that actually we weren't getting a strong immunogenic effect. That immunogenic effect was wearing off, 8 weeks later. And that means that the vaccine program has to be slightly altered. So instead of giving a single dose or 2 doses, you have to continually follow up with a vaccine in order to evoke that strong immunogenesis effect. But they're significantly more stable than live attenuated vaccines. So the advantages: reduced pathogenesis and easily produced, extremely stable, but they don't stimulate the immune system as strongly. You still need cold chain storage. There's a high risk for potential contamination because very much it's the same process as the attenuated viral vaccines, only the killing step is longer, and you don't necessarily gain long-term protection. So in this case, we isolate the live virus, we do a small purification of that virus, we establish an effective way of killing the virus, we evaluate the immunogenic potential of the killed virus, and then we do a large-scale production and clinical evaluation of efficacy. So you can see it's actually quite a long process for all of these vaccines. Subunit vaccines. you get several subunit vaccines. So a subunit vaccine is a part of the viral genome, but not the whole virus. So either it's the protein or the nucleic acid, which we'll come to in a few minutes, or it's the spikes of the proteins from the capsid of the virus. So subunit vaccines are parts of the virus. And of course, their major advantage is that much less potential for pathogenesis from these vaccines. So recombinant viral vector vaccines, these are vaccines that use chemically weakened virus to transport pieces of pathogen to induce an immune response. Viral vector-based vaccines present advantages over traditional vaccines in that they can enhance immunogenicity without the use of an adjuvant and induce robust cytotoxic T lymphocyte responses. Viral vaccines have been extensively studied in animal models, and many types of viruses have been developed as vaccine vectors and entered into clinical trials. The most promising methods for viral vector vaccines is prime boost strategies, where other vaccine types are used in conjunction with the viral vaccine. Viral vectors are mostly a promising system for developing safe vaccines for important diseases. And most commonly, viral vectors vaccines are applied in a therapeutic context. So your oncolytic vaccines, less common in a prophylactic environment, just because a lot of the viral vectors are ones that we have natural immunity against. So they have some advantages. Of course, high efficiency gene transduction, of course. Highly specific delivery of genes to target cells because you can decide which are their targets. You get both the humoral and the cell-mediated immune responses. Very good efficacy and safety. You can reduce the amount that you need to administer, and you can do quite large-scale manufacturing reasonably simply. But like I said, predominantly used in a therapeutic context as opposed to a prophylactic context. However, you run a risk of integration into the host genome, which can lead to other diseases. Pre-existing immunity to the vector can cause unwanted immune responses, and neutralizing antibodies can really strongly reduce the vaccine efficacy. In addition, viral vector vaccines are significantly more expensive than other types of vaccines and require much, much more characterization. But Merck is a leader in viral vaccines, mostly because we focus on viral vaccine production for vector-based vaccines for gene therapies. So in this case, we thaw and expand cells, of course, when we prepare our medium and inoculums. We grow our virus, harvest our virus, clarify again, DNA digestion, then we filter, we do chromatography, filter again, and then sterile filter, and then integrated fill and finish. So you can see this is quite a complex process, and you can see that 2 clarification steps is really where the expense of this process comes from. Recombinant protein vaccines. So there are 2 COVID-19 clinical trials on recombinant protein vaccines currently underway. Recombinant proteins or purified protein vaccines take the antigen from your pathogen, specific one, in this case, the spike protein. You express it in a heterologous expression system, bacteria or yeast, for example, and you can really produce a lot of protein. Then your immune system really produces antibodies to the protein antigen, protecting you from disease. And recombinant protein vaccines are based on the concept that humoral immune responses are targeted towards specific localized region of surface proteins, okay, known as antigens and epitopes. And that's really where this protein vaccine comes in. So you're creating very, very specific antibodies to very, very specific protein. Of course, that means that it's easily mutated out of, right? So if anything happens to that protein, then your immunogenicity is not going to work as well. Okay. So advantages, it's applicable in people with weakened immune systems. You get very long-lived immunity using a protein vaccine. You can get both humoral and cell-mediated immune responses, improved efficacy and safety. We know this can be engineered to present multiple antigens. So, for example, it's the polyvalent vaccine. Most famous polyvalent protein-based vaccine is the vaccines for HPV. So human papillomavirus vaccines have epitopes from, I think, 6 to 8 strains. And what that does is really allow for a single immunization to give you large amounts of protection, stable and low cost of production. But it has a low immunogenicity, of course, because it's only a single epitope. So you have to introduce it with a really strong adjuvant to get that initial boost to your immune system. You need multiple doses for long-term immunity, and neutralizing antibodies can really reduce vaccine efficacy. And again, you see there's a lot of similarities between vaccine processes, and you'll see there's a strong emphasis on filtration. So really process filtration and scaled filtration. And that's really where Merck is an absolute wonderful partner, because our research and development division, which I'm part of, we have small scale, but it's designed to be scaled up through our process solutions division. So what you use in the lab scale, you can use in production without having to change the technology underlying it. That's really where Merck's strength as a partner comes from. DNA vaccines, of course, this is where you put plasmid DNA into a person and it is the genome. Vaccines usually have 2 units, the antigen expression unit, and then the antigen encoding unit. So that is the promoter and the gene that you need in any kind of system, and then you need to have a way to select those plasmids. So antibiotics, for example. DNA vaccines are very uncommon, and the reason for that is just because they're very expensive and they don't really work that well. So again, this is predominantly used in a chemotherapy type setting. So we know that the advantages are it induces expression of antigens that more closely resemble native viral epitopes. And if you think about it, that makes sense. You're expressing the gene in the human cells, so it will be more similar to proteins that are expressed from the virus if it infects a human cell. Plasmid vectors can be constructed and produced quickly. That's a really strong advantage. And again, you can engineer multiple antigens into a single vector. Delivery is the major problem for DNA vaccines because your body will clear DNA much faster. Cannot be used to substitute your polysaccharide vaccines. And really you get high immune clearance. So you've just got to give such high doses that it becomes really, really difficult. So DNA vaccines, although an interesting technology, is not one that's been sort of widely adopted. And we're still working on really the delivery of these vaccines. And that's really the point at which they struggle. Merck, of course, we are partnered for plasmid-based vaccines. And again, you'll see this is a simplified process. You see it's got far fewer steps, and that's one of the big advantages. And of course, plasmid vaccines, you can actually use a single manufacturing capability or hub to produce these vaccines because they are not, because the DNA, you can easily remove it from the equipment. And what that means is that your capital investment allows you to produce more than one type of vaccine. mRNA vaccines are the holy grail of vaccines. They are most quick to produce. They are 8 of the 10 vaccines under consideration are mRNA vaccines. And the reason for that is that mRNA vaccines have a lot of the advantages of the other subunit vaccines. They're quick, easy to produce. You can produce more than one kind in the same system and all of that. But also mRNA vaccines have in other immunogenic vaccines because mRNA vaccines are not cleared by the immune system like DNA, and they're just much, much more effective in immune response than our protein vaccines. So production of RNA vaccines is laboratory-based, and you can use standardized and scaled productions, allowing quick responses to large outbreaks and epidemics. So the ability to produce mRNA vaccines is really critical to the function of a pandemic response because you can really quickly adapt existing infrastructure to respond. But there's a lot of work that still needs to go into producing standard treatments using mRNA vaccines, especially things like long-term efficacy. Because of how they work, it's possible that they don't induce those long-term immunogenic effects. So you get three types. You get non-replicating mRNA, of course, self-explanatory. It's destroyed when it gets turned into protein. In vivo self-replicating mRNA, so it makes copies of itself, okay? And then dendritic cell non-replicating mRNA vaccines. These are specifically for if you're trying to target the brain, okay? So mRNA vaccines, they all work like this, and they're very, very simple. You have a coding region or an open reading frame, a 5' region, and a 3' cap or the polyadenylation tail. And basically what happens is when you input them into the cells, they're immediately turned into proteins. And what that means is you get this really strong immunogenic effect really, really quickly, which is what makes them so interesting. So they're non-infective, so they're safe, even for immune-compromised individual. You get long-lived immunity. You induce both the humoral and cell-mediated immune responses. They don't integrate. You can engineer them for multiple antigens. They're very stable. They have an extremely low cost of production, and shorter development time makes them ideal candidates for pandemic preparedness. The disadvantages: unwanted immunogenic effects, delivery constraints, immune clearance, and processing and display. So those are still things that we need to work on for mRNA viruses, but really we've seen such a rapid progression of this technology over the last 6 months, and we'll continue to see that as large vaccine producers invest in developing technologies for mRNA. So just so you can see the benefit of an mRNA versus a traditional vaccine. So mRNA vaccines take 4 to 7 years for development. Traditional vaccines 10 to 15 years. The process is standardized, so it doesn't matter. You can produce 8 different mRNA vaccines and they've done it exactly the same way, whereas traditional vaccines, they're customized for each vaccine. The facility scalability, you just need one facility with bolt-on addition. So bolt-ons are just additional bioreactors for producing the template, but your fill and finish and your purification processing remains the same irrespective of your target, which of course is different from your traditional vaccine. Cost for developing an mRNA vaccine, 100 to 200 million dollars, whereas traditional vaccines, you're looking at anywhere between 200 million and $1 billion. Stockpiling, you can formulate fill and finish as needed, so you don't have to worry about expiry. And it's highly unlikely that you'll need an adjuvant. So those are the advantages and disadvantages of mRNA vaccines. So there are 2 processes for mRNA vaccines. The first is plasma DNA purification process and DNA linearization. That's just to make the mRNA. And then process 2 is just to produce the full product at the end. So it's a really, really simple product. And you can see this has 1, 2, 3, 4, 5, 6, 7, 8 steps versus 15 for your traditional vaccines. So it's really wonderful. So I hope you guys have really had some interesting interactions with this presentation. I hope I've given you guys something to think about. And I'd like to leave you with this thought from Nelson Mandela. And I think it really is our reason for looking at vaccines. Life and death in young children too often depends on whether he or she is born in a country where vaccines are available or not. And we have to start making sure that the future generations on the African continent are born in countries where we have the capacity to give them the best chance of a healthy life, and that is with good vaccine production. I thank you very much for your time and your attention.
Wow, wow, wow. Thank you, Dr. Samantha Nicholson. Oh, what an insightful presentation. You've increased our knowledge base and you've taken us through the challenges, the strategies of production, and the different technologies, for production. I'm very sure that as participants, we've gained enough insights, and our research and development process has been challenged this morning, this afternoon. And to look into our country's situation right now, because of our time, we have Dr. Chioma Ejekam in the house. Dr. Chioma Ejekam is a consultant public health physician and an expert in pharmacovigilance and pharmacoepidemiology with specialization in medicine risk identification and quantification. She's a fellow of the National Postgraduate Medical College of Nigeria and a member of the West African College of Physicians. She has over 12 years experience working closely with diverse stakeholders from the community, government institutions, bilateral stroke multilateral organizations in the field of public health with an excellent understanding of the health system. She is, like I said, an executive director with Bloom Public Health. And so she's here to give us more insights about the current situation of the vaccine process development and production in Nigeria. Dr. Chioma Ejekam, you're welcome. Thank you.
Thank you.
You have the floor. Yes, welcome.
Okay. Thank you. Just 1 second. Okay. Thank you and good afternoon, everybody. Thank you, Buki, for the introduction. And thank you, Samantha, for the fantastic and very robust, insightful discussion and presentation so far. So I'll just take us a few minutes now through what Nigeria is doing or has done so far and doing currently in the process of vaccine development. In August 2020, Nigeria made the first attempt or started a step towards developing the first ever Nigerian national vaccine policy. This is led by the Drug and Vaccine Development Division of the Federal Ministry of Health. And by the way, saying first ever, I think that doing our search, there isn't any African country with a vaccine policy. And in Asia, it's probably India. So we are not like doing badly here. So the policy document is actually intended to provide a broader framework to guide the creation of evidence base to justify the need for research and development, vaccine production, procurement, quality assessment of vaccines in Nigeria. And this document will address eventually the broad issues of strengthening the institutional framework, the processes, the evidence-based and framework required for decision-making for new vaccine introduction, like we have in the COVID period, to address vaccine security and program management and other product development issues. And of course, this, like we all have said, and Samantha, I believe, would have said that, but in Nigerian situation, the development of this policy is justifiable due to our immunization statistics. Like in 2018, the National Primary Healthcare Development Agency reported that the under-5 mortality was at 132 deaths per 1,000 live births. And over 4% of these deaths result from vaccine-preventable diseases. And similarly, in Nigeria, every day, approximately 2,300 children under 5 years die because of mainly due to preventable causes. So a national vaccine policy is required to achieve the effective manufacturing, storage, and distribution of safe and effective, good quality vaccines to our end users, especially as countries need to develop strong mechanisms to enable their decision-making and introduction of new vaccines and technologies in country. So in essence, efforts are in top gear by the Nigerian government to revive the country's once established vaccine production facilities through a recent, I won't call it recently, by the way, but more recently, through a joint venture agreement between the Federal Ministry of Health, that's Nigeria, with May & Baker PLC, which has given birth to a current company called BioVaccines Nigeria. I think Nigeria has a 49% stake and the May & Baker 51, but so this is one major achievement the Nigerian government has made. So just to take us a bit of history on the attempts at local production in Nigeria, vaccine production. Remember that from 1947, there have been efforts and eventual success of local vaccine production, but this was truncated in 1991. The Federal Vaccine Production Laboratory started in 1947, which went ahead to produce in Nigeria the smallpox vaccine, yellow fever vaccine, and the rabies vaccine. And the local production of the 17D yellow fever vaccine commenced in 1980s, but it got discontinued in 1991. But like I said, the good news is that this memorandum of understanding for the conversion of the once Federal Vaccine Production Laboratory to a private vaccine production and distribution company between me and Baker and the federal government of Nigeria has been formalized, and discussions are ongoing to kickstart and revive our local vaccine production. So we should always remember again that vaccine development, like I believe Samantha has rightly and robustly presented here, is a complicated, intricate process that requires several critical elements like funding. You need access to the market, access to technology, control systems, human resources, legal systems to back activities, power supply, very important in a Nigerian context, and management structures. And just to mention, in the vaccine development working group, I happen to be one of the co-leads for the vaccine manufacturing and the research and development thematic area for the vaccine policy development working group. And in the manufacturing thematic area, we are looking at human vaccines, animal vaccines, vaccine financing, vaccine production and export, vaccine quality regulation, and vaccine safety evaluation. And one of the major weaknesses towards vaccine development in Nigeria is the lack of national clinical trial centers or national clinical trials. And in the context of COVID-19 and vaccine trials, the last time I checked, the first vaccine trials in Africa started in South Africa, and the Uganda Vaccine Research Institute says they will be working towards starting a trial in December in collaboration with Imperial College London. I don't know if this has been updated like a month ago or something, but this is what's obtained as at August. So enough is not happening. Not enough is happening in Nigeria and Africa towards vaccine development yet. However, but we have opportunities. Nigeria has large and diverse population that provides optimal sample size for effective clinical trials. You must agree with me. And there's increasing donor presence and supports and other partners like Merck. And of course, this will present a positive economic outlook for Nigeria. However, threats, one major threat would be no robust policy to protect local vaccine manufacturing industries. The overbearing influence of multilateral organizations in vaccine-related policy decision-making in Nigeria. And of course, low literacy levels affecting acceptance of vaccine or clinical trial participation in Nigeria. But we are hoping that this policy, as part of our recommendation, will enhance and protect local content, local manufacturing, and export. In addition to developing a national legal framework to guide vaccine research and development and local manufacturing. And with this, we hope we'll make that very giant stride towards our vaccine local development. Thank you very much for having me.
Oh, thank you, Chioma. We're very grateful for that additional information that we're getting this afternoon. And so for us as Africans, we need to take the bull by the horn, at least take the giant step first, and then we will definitely get there. Thank you so much. And that takes us to the question and answer. Mr. Stanley Imadojemu to look at the questions that we have, and the presenter, Samantha, will give us the questions. Thank you.
Thank you very much, Dr. Olubukola. Thank you, Dr. Chioma, and also thank you, Dr. Samantha, for the wonderful presentations. We move straight now to question and answers, but just to let the participants know that during the question and answers, there's a poll chat that will be while you're listening, you can begin to fill in the poll. We want to get your feedback concerning the presentation that have just been given. So first question here is coming from Paul Akinduti. And the question says, what is the level of herd immunity to SARS-CoV-2 in Africa?
So this is still a really difficult question to answer, and that's because the serological surveys remain quite tough. And that's mostly associated with the fact that we aren't onto rapid diagnostic testing. We're primarily using PCR testing to evaluate the virus. But if we look at in terms of infection rates in the population, we probably are less than 15% saturation with antigen antibody recognition in the African population. And part of that is that we've actually been able to sort of restrict viral spread really well. So at the moment, difficult to estimate what our natural herd immunity is to the virus. I would say probably quite low, given our actually the fact that we've been able to it. So it seems a bit sort of counterintuitive. But until we have really good antigen-based screening, antibody screening tools, it'll be really difficult to establish sort of herd immunity in our communities.
Thank you very much. The next question is from an anonymous attendee. They say, why am I not discriminating? I am concerned. Why don't we have this collaboration also with Nigerian company? We have got a large population in need. And I think this is based on the reference you made to Merck's collaboration with, I think, a local player or something in Ghana.
Right. So we actually do have, we're in the process of engaging with several Nigerian, local Nigerian sort of entities around vaccines, as well as in Kenya, South Africa, and several other countries on the continent. And what we found is that there's a lot of willingness, but those partnerships are very, very much reliant on the availability of funding. So Merck is always here as a technological partner, and we will support where we can, but we have to also be sort of reticent of our investments. We need to make sure that our investments are being matched and that that we're going to see a return on our investments, because we are also limited in terms of our capacity. But certainly Merck is always open to relationships and developing relationships with partner sites across the continent. So there's always the opportunity to work closely with us and to really gain the experience of the Merck technologies and also the Merck scientific response. So feel free to engage with us. We're always looking for partners and for ways to collaborate.
Thank you very much, Dr. Samantha. This one is coming from Paul. He said, please, I would like to know how plant-made vaccines compare with those made in animal cells as regards effectiveness and costs. Did you get that?
Just repeat for me.
Please, I would like to know how plant-made vaccines compare with those made, probably he wants to say those made from animal cells as regards effectiveness and costs.
So plant-based vaccinology is actually super interesting, and it's a pretty new field. So we've seen it developed specifically for animal vaccines, so things like blue tongue virus, etc. And plants have the advantage, of course, that you can really produce a lot of plant material, so your production costs are lower. The concerns with plants are around epitopes. So plants fold proteins differently, and they alter proteins. So they do slightly different translational modifications on proteins. So the question always arises around sort of the immune activation effects of plant vaccines. But certainly the ones that are in sort of later stages of development, they have been shown to be sort of as effective as more traditional vaccine approaches, but their development process is a little bit more complicated and has to be evaluated really strongly, especially because plants produce proteins slightly differently. So there's codon optimization and so on that has to go on. But definitely protein vaccines in particular produced in plants are a growing field of interest, especially in tobacco as an alternative use for tobacco. So I think we're seeing a lot of it. We're mostly seeing plants in production of things like monoclonal antibodies, but I think we'll continue to see the development of that technology. So certainly there's an opportunity.
All right, thank you very much. This one is coming from Louis Ojukwu, and he specifically says this question is for Dr. Chioma. So Dr. Chioma, I hope you're listening. Louis is saying a while ago, certain vaccines like the rotavirus vaccine was covered under the immunization scheme by the government, free. But right now it is not covered, no longer free, and he probably wants to know why. Hello, Dr. Chioma there.
Okay, I'm here. Okay, so Louis, yes, I I know that certain information, certain factors actually inform the government to make decisions towards the vaccine that they will cover under that program. Because always remember, the vaccines, if vaccines are received free, but somebody is paying for the vaccine. But I am not aware that it's just there are certain areas that certain parts of Nigeria that still cover the rotavirus vaccine. I'm not aware that has been got off all over the Nigerian program right now. So we might need to confirm and get. I'm not aware it's no longer covered in the Nigerian program. But just to know that when there are decisions around vaccines that are taken or covering or not covering, depends. I'm cut off the prevalence of the disease in the community. But rotavirus covers the rare disease, which is very prevalent in Nigeria. I'm not aware has been taken off our program.
Thank you, Dr. Chairman, for that. Paul is also asking, Paul Akinduti, say, how effective is reverse vaccinology for development of SARS-CoV-2 vaccine compared to use if attenuated or killed cells? This is for Dr. Samantha.
So that's a really interesting question. We've seen a development of plasma-based therapeutics, which is usually the first step to reverse vaccinology. And what we've seen is actually we don't get a really robust immune response, even in people who've been infected. And what they're saying is that the immunity in those people is only effective for around 3 months. So at the moment, we don't know if we can reverse engineer the vaccine from sort of, the monoclonal or polyclonal antibodies produced by survivors. So at the moment, we don't know. We don't have enough data. But there are certainly a lot of studies evaluating reverse vaccinology for this and other pandemic diseases, because obviously plasma therapies are one of the first places we start to look for really robust interventions. So it's an interesting question. At the moment, we don't know, but I think if you keep an eye on the literature, we'll start to see that information filter through in the next couple of months.
Okay. A question here says, does Merck have any technical assistance program for companies interested in vaccine production in Nigeria?
Yeah, we do. So you can reach out to our team. We do an evaluation process, but certainly Process Solutions and our division of the business, we provide webinars like this and trainings around steps in the vaccine workflow, and a lot of technical assistance and questions related to our technologies. So we can certainly assist, although, obviously vaccines have to be produced in-house, but we can help, definitely.
Thank you very much, Dr. Samantha. I think that is all the questions that we currently have. We don't have any other ones, so I want to turn the program over to Dr.
Stanley,
yes.
Just to update Louis, okay? Now,
Okay.
I looked at some of our meetings. The rotavirus vaccine is being available as private vaccines, but the Nigerian government, part of the latest news is that it's going to be covered nationwide right now in 2020. So just to update Louis on the question.
Thank you very much. So Dr. Bukola, over to you.
Okay, thank you, everyone, the participants. Thank you for allowing us to enter into your space once again. Samantha, we're very grateful. Thank you for your time. Chioma, thank you. And then we have the CEO of Bloom Public Health in the house. Thank you for your presence. Thank you for your support. Mr. Stanley from Merck, we are very grateful for this collaboration. And Mr. Emeka, our ICT guy, you are always there. Thank you for all the support. And the next 2 weeks is another time with us. And we're going back to the lab. And we're going to talk about HPLC one more time. And this time we're looking at the tips, the tricks, and troubleshooting for improved LC performance. HPLC, the tips and the tricks and troubleshooting for improved LC performance. And we're also going to have a very experienced facilitator in the house, and the person of Candice Conyers. She is a chemistry field application specialist for the Africa and Middle East, also from Merck Life Sciences. Please keep a date with us. And for today, it's been very interesting. And we know that the global pandemic will continue to occur, just like we've had it in the past. And so as Africans, we have to get ready. We have to build capacity locally, particularly for the continent. And the cost to Africa cannot be calculated if she cannot find our own solutions. So for us as Africans, we have to be ahead and look at solutions to our problems. And like Samantha said, that the cost of it can be very, and from Chairman as well, yes, the cost is quite high, but then we have to take the right step in the right direction so that the vaccines can be available for every one of us to preserve the continent. Thank you very much, and very grateful for attending this seminar. Hope to see you in another 2 weeks, and thank you for participating in the poll. We've seen the result, and we promise that we continue to get better every time. Thank you. See you in another 2 weeks. HPLC tricks, tips, troubleshooting for improved LC performance. Thank you.
Thank you, Dr. Bukola.
Yes, thank you.
Maybe for those who still have questions, they can still push their questions forward.
Yeah.
We'll reach out to them through their emails that they provided after the program.
Okay.
Thank you.
Thank you. See you 22nd October 2020. Bye.